Hello, everyone. It's Mr. Cadell. We are in Chapter 16, DNA replication and the history of the discovery of DNA. Now, I like to do 16.1, the history of the discovery of DNA, in class with students and have them teach the rest of the class about all these famous names in the lead up to our understanding of DNA and how it functions, like Watson and Crick and Rosalind Franklin and all them. I don't know how that's going to work this year. Maybe we did it in class, and maybe you saw me present it. But anyway, that kind of bleeds a little bit into 16.2. And 16.2 deals with semi-conservative replication of DNA, which just means how DNA is replicated or copied. Why would we ever need to copy DNA? Well, science, I've heard wildly varying numbers on this, but science believes that you make about 8 billion new cells per day. And that's about 92,000 new cells per second. And in each one of those cells, with it having 6 billion nucleotides, or these A and T's and C's and G's, it's about 86,000 new letters per second put in the exact appropriate sequence and in the right place. And it's just it's mind-boggling to think of those numbers. It's going on right now in your body. So it's a process that we need to go over. And then once we're able to do that, you really understand DNA, we can do transcription and translation, which are in Chapter 17. You know, as I flipped through 16.2, there's a lot of information here, and it's all good. And I want you to read it all. Take your time. This is not an easy section of a chapter in this textbook. There's a lot of terms that you have to memorize. There's a lot of processes here. So again, do it well. Ask for help if you need to. It's a lot of pages, 7 or 8 pages long. And then eventually 16.3 is really small. 16.3 is just about how DNA, the anatomy of a chromosome, essentially, how DNA folds in order to make chromosomes, and how you can get all those 6 billion letters into something that's really small. In fact, if you were to take those 6 billion letters that exist in chromosomes in any one cell and stretch them out, they would stretch out to be about 6 feet, about as tall as me. But the nucleus, the diameter of a nucleus of a cell, is about 250,000 times smaller than the length of the DNA which fits in it. So that stuff has to really coil up, and they'll talk about that in 16.3.
Six years of graduate school, and look at our boy. Now, I remember somebody that wasn't too different from this not too long ago. Enzyme, what you doing today? I said enzyme. What you doing today? I said enzyme. Show me your body way, let me get to with some products. Enzyme. I can pull you on strength. I said enzyme. Cause I know that you can. I said enzyme. These letters also spell man. All we need to get is started. It's just a little bit of GTC. It's just a little bit of GTC. I can feel the evil. I can be your big star. You're the reason I'm being straight. It's fine. You have come a long way, but in my way, the man's are higher today. I said it's fine. Now it's time to go back. We have got to increase the wood inside. I think I found what I need.
Okay, that's awkward. Let's move on with my favorite dance move. Which is your favorite dance move at the dances that we have here at Battle Mountain? And mine, of course, is the double helix. Awesome. So if you're sporting that one along with the shopping cart or the sprinkler, I don't know what the other one was called, at the next dance, extra credit, or your friends will hate you forever. Remember, this is something that we've learned in the past. The nucleotide polymers are linked together to build a polynucleotide. That just means we have this nucleotide which exists of a sugar, phosphate, and a nitrogenous base. That's that unit there on the right side of this picture, and they're linked together. You make then these long chains. This might be one side of a DNA molecule, and it could be hundreds of millions of letters long without a single break. The key here is that you can count the carbons, as we've done before, starting at noon or 12 o'clock and going in a clockwise fashion, and count the carbons, and then you see where the number 3 carbon is. They call it the 3' carbon, and then you can see the 4th and then the 5th 5' carbon, which we have yellow arrows pointing to here. But what I want you to remember is that DNA is actually structured anti-parallel. All that means is that these strands are parallel one another, but they run in opposite directions. Does anyone remember why they run in opposite directions? You can see there how it's kind of like upside down. The sugars, the ribose sugars are upside down, and then the nucleotides themselves are upside down. And the reason why is because you can only add nucleotides to the 3' end of the growing strand. You can see that here. And that's going to cause all sorts of problems and be the kind of the minutia or nuance of this 16.2 that we're going to have to spend a lot of time on. So therefore, new DNA can only elongate in the 5' to 3' direction. And you see up here I put a highway sign, 53, and say it with me now, 5 to 3, 53. I just want you to commit that to memory, 5 to 3. So you can only continue to add on the 3' end. So it keeps going in the 5 to 3 direction. Well, let's do this. Go ahead and take a piece of paper and draw these two sides of a DNA molecule here. And that's representative of this up here. And then we're going to split that open in order to replicate this down here at the bottom. So let's imagine if we did add to this. And we could say that on the original strand there was an A and a T and a C and a G and so on. And then you could say, oh, that's easy. I'm going to put a T here, and then an A here and a G and a C. And we could then even say, okay, of the new strand that's being added, complementary to this strand in a semi-conservative fashion, this is the 5' end. You could say, oh, this is the 3' end. And oh my gosh, it's super easy here. And as we go up here and this fork continues to open, we just keep adding in this way. Because we can add to the 3' end. It's not a big deal at all. What's interesting is that if I say that this side, this is a T, an A, a G, and a C, then I know what's here. This is a T, an A, a G, and a C. The point is we're just trying to mirror with our new strand what was on the old or parent strand, because eventually this whole thing's going to unzip, and then we're going to get 2 strands of DNA where we just originally had one, and they're going to be exactly the same as one another. I'm just going to erase this. So that's good and that's easy, no problem. But what do you do here? If I say then that this corollary to this new kind of daughter strand is facing this direction, the 3' end is here, and I get it that an A should go here, but you can't add an A there. You can only add to the 3' end. You can only keep adding where you see this. And so if I can only add here, I can't add here. So how do I do this? And again, we're going to go over this, and it causes all sorts of kind of wrinkles here with DNA replication. So I guess I'm just going to leave that blank for right now, but we'll get into this detail. Well, when DNA replications happens, and I already told you how often it occurs, there is a lot of accuracy that must be done here. In fact, I read that the enzyme that facilitates this DNA polymerase makes a mistake about 1 out of every 100,000 letters. And you can imagine if they're doing 86,000 letters per second, how quickly in a single cell, how quickly that must be occurring. It's incredible how accurate it is. You do have these DNA repair enzymes which go through, and then they are able to identify when a mistaken nucleotide has been put in sequence. So imagine, if right here, if instead of this G, we put like an A, well, these enzymes will come through here once this is all done, and they'll be like, okay, this is the parent strand, and this has this, and then this has this, and that doesn't match. And so there's different ways and they'll talk about it in your book, and we'll watch some movies on it eventually. There's different ways that they will be able to cut literally this incorrect letter out and then put the correct letter in. And they're very good at this. In fact, I read that as a result of this, you only have like one mistake every 1.5 billion letters, which is a few mistakes per cell. But I've also read something totally opposite, and they say that you only give to the next generation, like let's say in your eggs and sperm, when you make a baby, about 100 stable mutations where this was an incorrect letter, and then it stays that way. And then you may have some sort of misshapen protein or something that's created that you didn't originally have as a result of that. Now that could be good in the sense that if that misshapen protein gives you an advantage in your environment, maybe it works better, maybe it does something totally different, then if you're able to reproduce as a result, reproduce and then pass it along to your offspring, that's what we call natural selection advantage, or maybe it's the fuel for evolution eventually. So it's not always bad that this happens, but often it is bad. A lot of diseases occur when these kind of stable genetic mutations kind of get locked in and then they get passed along to future generations. Well, you notice that this picture is something similar that you saw in chapter 12, which is DNA replication for mitosis, and then also it occurs in meiosis. Well, let's talk about bacteria. So one of the pictures, I think it's, I forgot the name of the figure, the number of the figure, but in bacteria, what happens is that you get DNA replication of this big circular chromosome that exists in the middle of the bacteria, and you get these things called replication bubbles, and they're caused by origins of replications going around. So the idea is that it's a circle. You start at one place, and then these DNA polymerase enzymes go in opposite directions, and then eventually you can see it here that they will kind of just zip out the brand new piece of chromosome here. And so it's almost like a zipper. I'm going to show you a movie in a second on it. Just understand origin of replication, that's where it originates, and then it goes in two opposite directions, and then you have a brand new circular piece of DNA where you once had a single circular piece of DNA.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
So that's great, and that is very similar to what happens in eukaryotic cells that have these linear or non-circular chromosomes. They're like a line, right? In eukaryotic cells. However, here's how we do it. And when I tried to throw out those numbers and impress you earlier, the idea is that there isn't a single enzyme doing 86,000 new letters per second. What happens is that you have many thousands of origins of replication replicating the DNA, maybe even on the same chromosome. Chromosome 1 has 134 million letters bases on it. And the point is that there could be many of thousands of these DNA polymerases copying the DNA. And what happens, I'm going to show you in the next picture, is that they actually, all that unzipping and copying like this, they kind of run into each other. And then all of a sudden, by default, you have two segments of brand new DNA. Let me show you what this picture looks like. So you can see it here. This is also a picture in the book. There would be the replication fork, or the origin of replication. We call this maybe a bubble. And you can see how they're getting bigger here, and eventually they're just going to kind of run into one another. So that's how it happens so quickly in every one of our cells. It says the replication proceeds in both directions from each origin until the entire molecule is copied out. It's pretty cool. Well, what the rest of 16.2 deals with is the detail. So if you look on the picture and you see kind of there's a fork there, maybe the fork kind of looks like this, and we're going to spend a lot of time with definitions of enzymes and what really happens here, what it's called. So this is a picture of what it looks like from your textbook, a cartoon form of it. I've got it drawn up here on the board. At the end of each of these bubbles, or one side of this bubble, is what we call a replication fork. It's a Y-shaped region where new DNA strands are elongating. Okay, that makes sense to me. It's kind of like a zipper. And let me show you this movie. It's a few years old, but I think it does a good job.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helix. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules. It's similar to copying information on a computer. Hundreds of megabytes of data can be backed up in minutes by running a copy onto a CD. The copying process is accurate, typically less than one mistake in a billion. The DNA copying process in our bodies is even more accurate, 1,000 times better than most computers. Remarkable biochemical systems have evolved to double-check the new DNA strand and fix any mistakes.
Oh, did you hear what she said? A CD. Do you even know what that is? That little silver thing. Anyway, here are the players. You got to know these. I would put a star next to this picture, table 16.1 in your book. You have to memorize these. You got to know what they are and what they do in this process. There's no way around it. We're going to touch on each of them right now. So the first one, topoisomerase, is actually an enzyme that exists out ahead of the replication fork, or maybe it is the beginning of the replication fork. And I'm holding this piece of yarn here because I don't know if you can see this, but you can imagine if this thing is a helix, right? It's all spun like this. And imagine if you were to take any sort of rope or something that's spun around itself and you were to pull it like this. What happens is that as you open it in order to then replicate the insides of this, you get this kind of bunching and curling of all this tension on the front. And what topoisomerase does is it breaks the DNA, unswivels that tension, and then re-anneals the DNA to itself, and then it just keeps doing that ahead of the replication fork as kind of some tension gets put onto it. So the next one is helicase. And what helicase does is it actually is the thing that unzips the DNA. And you saw it there, unzips it very quickly. There are enzymes that untwist and unzip the double helix. So they break those hydrogen bonds between the nucleotide complementary bases there. A couple years ago on Valentine's Day, I got a Valentine from Jane Goodall. Anyone know who Jane Goodall is? She's a chimp researcher from Kenya, who did a lot of great science work long ago. And of course, I open it up and it says, "Hi, Tim, if I could be any enzyme, I'd be a helicase so I could." Happy Valentine's Day. And I don't know if that's appropriate, but anyway, I covered my mouth. Hey, thanks, Jane. Appreciate that. So the single-stranded binding protein is another enzyme that exists here. And what it does is it acts like a sleeve or a sock, I think. So once the helicase opens up the DNA, it definitely wants to the zipper wants to kind of come back together and create those hydrogen bonds again. But what these things do is they serve as something that stops that from happening. They're like a blocker, essentially. They bind to and stabilize single-stranded DNA until it can be used as a template, meaning for the next daughter strand of DNA that comes in there. The next complementary strand of DNA in semi-conservative replication. And then I guess this whole new thing would be called a daughter strand of DNA, both sides eventually, versus the parent strand of DNA. Like a sleeve. The enzyme then called primase, it's going to start an RNA chain from scratch, and it adds RNA nucleotides one at a time using the parental DNA as a template. So what you get here is this actually, I'm just using a different color because this is RNA primase. And what it does here is it will add the complementary letters. So this will be a T and this will be an A and so on. But it's 5 to 10 letters. And the whole goal of this RNA primase is to give a 3' end. So I'm just drawing a 3 there. And when it gives a 3' end, then it gives the next guy, the DNA polymerase, the guy who adds about 50 letters per second, going in this direction, for instance, it gives it an end to add on to. So you have to give it an end because there is no end. When this thing starts, there's no end, there's no 3' end here for things to be added to. So this is a piece of RNA that gets added on here, probably because RNA is single-stranded and it works fine. But then this has to be changed to DNA. So this is going to have U's where there once was T's. It's also going to have a different sugar. It's going to have ribose sugar versus deoxyribose sugar that the DNA strands have. So those two things have to be changed here eventually. This is just an important thing called RNA primase. It's shown in red here on the picture. Primers is short, 5 to 10 nucleotides long, and the 3' end serves as the starting point for the new DNA strand. You can see that I haven't touched this yet because it's what we'll get to next. Finally, DNA polymerases are the things that start adding all these letters really fast. And on this side, it's going towards the replication fork as that fork is opening up towards the ceiling. They add new nucleotides to the 3' end, no problem. It also happens on this side. The DNA polymerase works as well, adding nucleotides to the 3' end. But as you might be kind of getting the picture of now, especially from that movie that you saw, it's going to be added in this direction. And I'll show you how we do that in just a moment. So this is kind of the bigger picture that you'll see a couple pages later in the chapter. You can see the primer there. You can see the DNA polymerase 3, which has been adding the DNA in sequence in a 5' to 3' direction. This would be on the top strand or on the right-hand strand here, because this is a 3' end. This is also a 3' end, which allows us to do a 5' end and add towards the replication fork. You can see there at the bottom kind of a hint also that they're kind of done in chunks. It's kind of weird. You can do that in a second.
Using computer information based on the latest research.
So what I want you to notice here is that we can see the helicase unzipping the DNA from the left-hand side. You can then see all these other enzymes doing their job, but you can see what they call the leading strand going off to the bottom or to the right, and then it's done all at once. But then the other side is kind of done in chunks, and it's done going backwards, going towards the replication fork. It's pretty cool how all of these do this all day long, even though when you're not thinking about it. Okay, so let's get to a little bit more detail once you teach someone about what you know so far. So go ahead and press pause, please. Well, let's keep going. So I've alluded to this on the 3' end, on the right side on this picture, super easy going towards the replication fork. But what do we do here? Well, here's what we do. So right side easy, again, right side easy. Or up top of this, I guess it depends if you were kind of laying on your left side, it would look like the right side if you were looking at the screen. Anyway, that's called the leading strand, right? It's super easy. But the guy who figured this out, his name was Okazaki. He's a Japanese researcher. You can again see the replication forks here. And he figured out that on the other side, the DNA was copied in kind of a backwards fashion away from the replication fork. So the point is, as this fork opens, you get like small chunks of DNA here, and then a small chunk of DNA here, which is complementary to this parent strand of DNA. And then as the fork continues to open, you get more here and then you get more here. And as the fork opens up towards the ceiling, you get these little chunks, and you can see that on this picture. To elongate the other new strand, called the lagging strand, the DNA polymerase must work in the direction away from the replication. You can see that there and see that. So we're going to go to this picture in the book. So here's what happens, is that the RNA primer will come on, and it will create, so this is the 3' end. So let's imagine that we're here. So this RNA primer comes in, it creates a 5 to 3' RNA primer of 5 to 10 nucleotides, and then and only then can the DNA polymerase come here and add to this end. Now this will, like I said, this will eventually get replaced with DNA, but eventually we're going to hit this end here. And so then what the primase does is that it will come in and will add another RNA primer. I'm just writing 5 to 3 here, and then we can add another bit of DNA. We keep doing that in the opposite direction of the leading strand. This is called the lagging strand because it literally takes longer to copy this side semi-conservatively. It just takes longer, and it's lagging in DNA replication speed. You can see this thing called the Okazaki fragment. That's that little piece of DNA that's made as a result of the primer being laid down. You can see the enzyme that's popping off, and it's basically leapfrogging backwards here. That would be the DNA polymerase III enzyme, which is laying down all the new nucleotides. The lagging strand is synthesized as a series of segments. See that, called Okazaki fragments. See the primers there in the middle. DNA polymerase I is the thing that's going to come in, and it's going to actually take away all these RNA nucleotides and create DNA where RNA once was laid down as a primer. That's DNA polymerase I's job. Remember, it's going to change a U to a T, and I said it's also going to change the sugar from a ribose sugar to a deoxyribose sugar. And eventually, there's actually like a little gap between where the RNA polymerase was and then the DNA. So sorry, the RNA polymerase is going to be replaced with DNA, but then we have to bind that to the other DNA that's already been laid down. And there's a new player here, an enzyme called DNA ligase. It's going to ligate or marry two things together. You're going to see that. The lagging strand is synthesized as a series of segments called Okazaki fragments, which are joined together by DNA ligands.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
You can see that evidence on this picture. This is a picture from your textbook. Make sure to really start to focus on what you're seeing here and how all these enzymes play a role. I'm going to show you another movie on this But first, let's pause and teach your neighbor.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, sweet. If you need to do this again, press pause now that you've seen that movie and go over these again, please. It's going to benefit you in the long run. Well, I do have to tell you one thing about DNA polymerase 1 and DNA ligase. So this is kind of like the dirty secret here. So if we have an RNA polymerase that is laid down a 5 to 10 nucleotide RNA primer, that's fine. The DNA polymerase 3 can come in here. This is the leading strand, and everything's fine. It's just adding nucleotides. But the problem is this. There is no, I'm just going to write a 3' with a question mark here. There is no 3' end right here. It's just kind of floating. There's nothing for the DNA polymerase 1 to jump on to a 3' end and then get rid of these. It just can't do it because it needs a 3' end to grab onto, and there isn't one. So let me erase that. And so what happens is every time a cell divides, you lose a piece. So what happens is that this is RNA. So it's not going to do anything when you go to try to make do transcription and translation and make proteins. So this piece of the DNA gets cut off every time, and that's okay because so this is called a telomere. This space on the DNA that gets cut off every time. It has like this sequence TTA, AGGG, I think it is. It'll tell you in your textbook, and it's just repeating probably 50 times or 100 times or whatever. And every time the cell divides, that's fine. That gets cut off because it's a nonsensical sequence of letters on DNA. It's not going to code for any proteins. And the reason why I say it's okay is because there's no problem there, but there's also a good thing, and that is that what that means is that eventually the cutting off of this bit of DNA will get to the good stuff way over here, and then the cell will just say, okay, gotta do apoptosis. Things aren't going well. I don't want to get rid of this good sequence of DNA. So the cell will basically kill itself. And that's fine because what happens is every time you divide cells, mutations have a potential to pile up. And so again, eventually you want these cells to no longer divide. So there is an enzyme called telomerase, which will keep extending this telomere back that direction on the black strand up here. And that happens in our germ cells or sex cells in the ovaries or the testicles. And also it happens in cancerous cells. What cancerous cells do is they can divide indefinitely a lot of the time because what happens is the telomerases keep adding bits of this telomere back on here, and so they can basically divide in theory forever because they never get rid of this piece, this kind of weird piece that is a DNA attached to an RNA. That never happens over here where the good stuff is. It's pretty cool. Read about telomerases in your textbook. Some people say it's the fountain of youth. If you can elongate your telomeres, you will live longer because your cells will be able to divide longer for a longer amount of time, and you'll be healthier. But maybe you won't be healthier. I don't know. Make sure that you spend a lot of time with these guys. Thank you for your attention.